Hello and welcome to this GCSE Science Core Biology revision video on variation. In this video we will cover inheritance, types of reproduction, cloning and genetic engineering. Inheritance refers to inheriting certain genes from each parent. Alleles are different forms of a gene. They can be either dominant or recessive. You inherit one full set of genes or chromosomes from each parent. Some key points to consider are that genetic information is in the nucleus of each cell. Inside the nucleus are chromosomes that are made up of DNA. Genes are small sections on the DNA and control enzyme production and protein production, and genes are passed to you in the sex cells or gametes of your parents. There are two main types of reproduction: sexual reproduction and asexual reproduction. Genetic information passed from parent to offspring is contained in genes carried by chromosomes in the nucleus. Sexual reproduction produces offspring that resembles their parents but are not identical to them. Organisms have sex cells called gametes. In human beings, the male sex cells are called sperm and the female sex cells are called ova. Sexual reproduction happens when the male gamete and the female gamete join together. This fusion of gametes is called fertilization. Sexual reproduction allows some of the genetic information from each parent to mix, producing offspring that are similar to their parents but not identical. This leads to variation. In asexual reproduction, you only need one parent. Since there is one parent, there is no fusion of gametes and no mixing of genetic information. As a result, the offspring are genetically identical to the parent and to each other. In other words, they are clones. Clone is where we go and make a genetically identical copy of another organism. When cloning plants, there are two ways. The first way is cuttings. During cuttings, you remove a small section of the plant. New roots and shoots will form to give you a new plant. You may need to put plenty of moisture and rooting powder on this to make the process happen quickly. The advantage of cuttings is that it's quick, easy and cheap. You produce genetically identical plants. This is commercially used by industries such as fruit growers. The second method is by tissue culturing. This is where you take a small part of the plant and you break it down into individual cells. You put those individual cells onto some sort of agar and they'll grow into genetically identical clones of the plant that you took that original leaf from. It is expensive, but it allows thousands of new plants to be grown from one tiny piece. You need to use hormones to make the small group of cells produce a big mass of identical plant cells, which guarantees that you will have the desired characteristics of the plant that you start with. In animal cloning, there are two main forms. The first form is embryonic cloning. In this, the best cow is given fertility hormones to produce lots of eggs and fertilize from the best spawn. Each egg divides into an embryo of individual cells. Those cells are then separated and grown into individual embryos. These fully grown embryos are then implanted into surrogate mothers. The second type of cloning is the same as what was used to clone Dolly the sheep. So, if we want to clone sheep A, you extract a skin cell from it. From that, you extract the nucleus. From a female, you extract an egg cell, you remove the nucleus from it. This leaves an empty egg. The nucleus from sheep A is then inserted into the egg and fused to it by giving it a small electric shock. The egg is then allowed to grow into an embryo, which can then be implanted into a surrogate mother. A few months later, a genetically identical sheep to sheep A should be born. The advantages of adult cell cloning is that it can save animals from extinction, and you can clone genetically engineered animals that are medically useful. The disadvantages are all down to ethics. In genetic engineering, we are looking at changing the DNA of an organism. The example that I'm going to go through is genetically engineering bacteria to produce insulin, but all forms of genetic engineering require you to talk about taking the DNA out of one organism and inserting a gene into it that codes for a particular feature or protein that you want to produce. So, we start off by identifying the human cell that we want the gene out of, in this case a pancreatic cell. The DNA is extracted from it and the gene is located. The gene is then cut out using restriction enzymes. At the same time, a plasmid from a bacteria is removed and cut open, also using a restriction enzyme. The gene is then inserted into the plasmid using other enzymes. The plasmid is then reinserted back into the bacteria. The bacteria is allowed to reproduce, and as it reproduces, it will produce insulin. To finish off, a few revision tips. Remember to turn off any distractions, and quality revision is better than quantity.